Hello and welcome to my YouTube channel, my remote class. Today's topic is Krebs cycle. Before going through Krebs cycle or citric acid cycle, let me tell you the history first. Albert Saint Gregory established components and reactions of citric acid cycle in 1930s, and citric acid cycle was finally identified by Hans Adolf Krebs and William Arthur Johnson in the year 1937, and both of them were working at the University of Sheffield. In the year 1953, Krebs got Nobel Prize. The citric acid cycle is also known as TCA cycle because there are 3 carboxylic acid groups present, and it is also known as Krebs cycle, named after Hans Krebs. Krebs cycle is extremely important because it is a common pathway of carbohydrate, fat and protein metabolism. Now let us see where does it take place. In prokaryotic cell, it takes place in cytosol, and in eukaryotic cell, it takes place in the matrix of mitochondria. Now, what are the purposes of Krebs cycle? Krebs cycle can be used for multiple purposes. Number 1, it is used to generate energy through oxidation of acetyl coenzyme A. Secondly, it is used for synthesizing NADH. And thirdly, it is used for the production of amino acids. So let's see what happens in Krebs cycle. Now these are the reactions of citric acid cycle or Krebs cycle. Before going through the explanation of citric acid cycle or Krebs cycle, let me recall some of the earlier things which happened. Here you can see at the top the glucose is written. Now from glucose, what happens? Through glycolysis, 2 molecules of pyruvate generated. And from 2 molecules of pyruvate, acetyl coenzyme A is formed. So we can write it down like this, that in glycolysis, 1 molecule of glucose gives rise to 2 molecules of pyruvate. This process is glycolysis. Glucose is a 6 carbon compound, whereas the pyruvate is a 3 carbon compound. Now, from pyruvate, acetyl CoA is produced. This happened due to the pyruvate dehydrogenase complex. And during this, what happens? 1 molecule of NAD plus removes 1 molecule of NADH. At the same time, it also removes 1 molecule of carbon dioxide. So as the carbon dioxide is removed, so there is reduction in the number of carbons. As a result, from 3 carbon to 2 carbon acetyl coenzyme A is produced. So here is the acetyl coenzyme A. Now as you can see that oxaloacetate is here, which is a 4 carbon compound. It is reacting with 2 carbon acetyl coenzyme A to form 6 carbon citrate. So here is 4 carbon, here is 2 carbon, and both togetherly making 6 carbon citrate. From citrate, isocitrate is formed, which is again 6 carbon compound. And from isocitrate to alpha ketoglutarate is formed. When this is forming, as you can see that 1 molecule of NADH is removed and 1 molecule of carbon dioxide is removed. So as a result, what happens? Once again, there is reduction in the number of carbon, and from 6 carbon to 5 carbon alpha ketoglutarate is formed. Now from alpha ketoglutarate, succinyl coenzyme A is produced. When this conversion is taking place, once again 1 molecule of NAD plus is giving rise to 1 molecule of NADH, and at the same time 1 molecule of carbon dioxide is removed. So once again there is a loss of carbon. As a result, what happens? From 5 carbon to 4 carbon, succinyl coenzyme A is produced. Now, from succinyl coenzyme A to succinate is produced. When this conversion is taking place, GDP is forming GTP. What is GDP? GDP stands for guanosine diphosphate. And what is GTP? GTP stands for guanosine triphosphate. Now from succinate, what is forming? Fumarate is forming. And in this reaction, we are finding that FAD is giving rise to FADH2. And from fumarate to malate is produced. And here one molecule of water is required. And from malate to once again oxaloacetate is forming. And when this conversion is taking place, then NAD plus is giving rise to NADH. So these are the reactions taking place in Krebs cycle or citric acid cycle. Now let us see which enzymes are involved. Now I am writing 1, then here 2, then here in this step 3, here in this step 4, here 5, here 6, here 7, and here 8. So let us start with number 1. Number 1 is citrate synthase. Citrate synthase. Number 2 is aconitase. Number 3 is isocitrate dehydrogenase. Number 4 is alpha ketoglutarate dehydrogenase. Number 5 is succinyl-CoA synthetase. Number 6 is succinate dehydrogenase. Number 7 is fumarase. And number 8 is malate dehydrogenase. So these are the enzymes which are involved in various steps of citric acid cycle. Now let us calculate the number of ATPs produced in citric acid cycle. Before going through the calculating the number of ATPs, let us consider how many NADH are formed. Here is one NADH, here is another NADH, here is another, and here is the last one. That means if you count, it is 1, 2, 3, and 4. So number of NADH is 4 Now let us calculate how many FADH2 is formed. Just one FADH2 is formed. And let us calculate how many GTP is formed. Just one GTP is formed. Now, one NADH, as per old school, gives rise to 3 ATP. Whereas, one FADH2, according to old school, gives rise to 2 ATP. And one GTP gives rise to 1 ATP, because this is the direct one which is producing ATP. This is a substrate level oxidation. So it will be 4 into 3, that means 12 ATPs. This will give rise to 1 into 2, that means 2 ATPs. And finally, the last one will give rise to 1 ATP. So if you sum up all of them, it will give rise to 15 ATPs. So 15 ATPs are formed from one cycle. And for one molecule of glucose, this cycle runs twice. That means 15 into 2, it will be 30 ATPs. Now what happens to modern theory? Nowadays, the calculation is a bit different. What is this calculation? One NADH gives rise to 2.5 ATPs. And one FADH2 gives rise to 1.5 ATPs. But the value of GTP remains unchanged because, as I said, it's a direct method or direct process, substrate level oxidation. So let us calculate in this new way. It will be 4 into 2.5. That means 10 ATPs will be there. And here 1.5 into 1. That means it will give rise to 1.5. And here is 1 into 1 will give rise to 1. So if you sum up all of them, it will give rise to 12.5. So 12.5 ATP is generated from one cycle. But as I said, for one molecule of glucose, the Krebs cycle runs twice. That means it will be 12.5 into 2 equal to 25 ATPs generated in Krebs cycle. I hope it clears all your doubt as far as the citric acid cycle is concerned. If you like my video, then do like, share and comment. Thanks for watching my video.